Let us start with the first law of Mendel, that is, law of dominance. According to this law, that is, law of dominance states that out of, or rather when, two different alleles for a character occur in an organism, then only one of the two alleles expresses itself. And so the allele that expresses itself is called dominant allele, or we can say dominant gene, while the other allele that does not expresses itself is called recessive gene. Now, let us understand this law with the help of an example. As said earlier, that is, let us take the previous example where the T and T, that is the tall and the dwarf plant, they are crossed with one another. That forms three tall plants, that is this, and a single dwarf plant. Now, this was about the F2 generation, while in F1 generation it was when T crossed with T gives therefore, when a tall plant was crossed with a dwarf plant, then the resultant F1 generation was only tall plant. Because of this, the gene or the allele that expresses the tall plant is considered as the dominant gene, while the dwarf plant that does not express itself in presence of tall plant is called recessive gene. And therefore, here in this case, T represents the dominant gene, while small t represents the recessive. And so, on this basis it was derived that the F2 generation plants are always in the ratio of 3:1, where the dominant character expresses the most and the recessive the least. Based on such a result, Mendel derived this law of dominance. Moreover, it was on this basis that when Mendel chose different characters of the plant, on that base the dominant and recessive trait was obtained. In case of seed shape, when crossing between round and wrinkled, round was considered as the dominant and wrinkled as the recessive. Similar was for the seed color, where yellow was dominant and green was the recessive. In similar manner, different characteristics possess different dominant and recessive traits, as a result of which the experimental results were obtained in all cases as 3:1. Then came the second law, that is law of segregation. According to this law, any gamete that contains only one gene for an expression of a character. And therefore, on the basis of this law, that is the second law, law of segregation, any gamete, in order to express a character, only one gene was responsible. And since only one gene was responsible, it was also known as law of purity of gametes. Thus, it was on this basis that a pair of contrasting characters, that is in terms of height, the two contrasting characters was tall and dwarf. When they were brought together in hybrid, these two factors or alleles although remained together but did not got mixed with one another, and therefore in this manner, when the gametes were formed from each other, only one entered each gamete. On this basis, it was calculated that an organism can be either homozygous or heterozygous depending on the offspring that was obtained. Homozygous means the two similar traits, that is, two capital T that depicts the tall, similarly to small t that depicts the dwarf. On the contrary, heterozygous means two different traits for a characteristic, that is, that depicts the tall plant. Out of which these two, one trait is responsible for the tall and the other is for dwarf. So although the combination of two resulted into tall plant, but still it is termed as the heterozygous character. And therefore, although in heterozygous mode, its gamete will always be pure for a particular expression of that character. On the basis of law of segregation, there was another type of cross that was understood, that is test cross. This cross is arranged for deciding whether a plant is homozygous or heterozygous. That is, test cross can be defined as a cross arranged for deciding whether an organism is homozygous or heterozygous So, here in this case of example, that is tall and dwarf plant of the pea, if we consider a homozygous tall that on multiplication with a homozygous dwarf results into the formation of one homozygous tall plant, two heterozygous tall plant and one homozygous dwarf plant. So, in this manner, the ratio of homozygous is to heterozygous was almost equal, that is 2 homozygous and 2 heterozygous. Although the phenotypic ratio is more of tall plants than that of dwarf plant. Similarly, if the heterozygous tall is multiplied with the homozygous dwarf, then it results into the formation of 2 heterozygous tall and 2 homozygous dwarf. Thus, this was an exact and appropriate ratio of 1:1, that is 1:1 for dwarf and tall plant. That is, there were equal proportion of tall and dwarf plant. Therefore, on this basis, that is on the basis of this cross, genotype of the plant can also be determined. In case of test cross, generally the ratio obtained is always 1:1.